Hi, welcome to my YouTube channel BioExplorer. I am Dimutjaratna. This is an educational channel and this channel will be very useful to improve your biological knowledge. Today in this video, we are going to talk about enzymes. What is an enzyme? An enzyme is a macromolecule which acts as a biological catalyst. They are usually proteins, though some RNA molecules act as enzymes too. Enzymes are produced in living cells. Most enzymes are heat sensitive. They lower the activation energy of the reaction they catalyze and increase the rate of reaction. They do this by binding to a substrate and holding it in a way that allows the reaction to happen more efficiently. Activation energy is the amount of energy that must be put in for the reaction to begin. The reactant molecules where an enzyme will bind are called substrates. The part of the enzyme where the substrate binds is called the active site. The matching between an enzyme's active site and the substrate isn't just like two puzzle pieces fitting together. Enzymes are highly specific to the substrate. Most enzyme-catalyzed reactions are reversible. The enzyme activity can be affected by a variety of factors such as temperatures, pH, and concentration. They are not being used up during the reaction. Some enzymes need non-proteinous components to catalyze the reaction, which are known as cofactors. The mechanism of enzyme action. The enzyme binds to its substrate, forming enzyme-substrate complex. While enzyme and substrate form their complex, catalytic action of the enzyme converts the substrate to the product. The substrate binds to a specific region of the enzyme. This region is called the active site. The active site is formed by only a few amino acids. The set of amino acids found in the active site, along with their positions in 3D space, gives the active site a very specific size, shape, and chemical behavior. The active site is uniquely suited to bind to a particular substrate and help them undergo a chemical reaction. Other amino acids in the enzyme are needed to maintain the shape of the enzyme molecule. Some enzymes accept only one particular substrate and will not catalyze a reaction even for a very closely related molecule. Other enzymes can act on a range of target molecules. The shape of the active site is complementary to the shape of the specific substrate of the enzyme and hence important in the substrate specificity of the enzyme. The specificity of an enzyme results from its shape. The shape of the active site of an enzyme is not always fully complementary to its substrate. As enzymes are not rigid structures, the interactions between substrate and active site may slightly change the shape of the active site so that the substrate and the active site become complementary to each other, resulting in an even tighter fit. This adjustment of the enzyme is called induced fit mechanism. The tight fit not only brings the substrate molecules and the active site close to each other, but also ensures the correct orientation of the molecules to help the reaction to proceed and catalyzes the conversion of substrate to product. Thereafter, the product departs from the active site of the enzyme. The enzyme is then free to take another substrate molecule into its active site. In all cases, the enzyme will return to its original state at the end of the reaction. When an enzyme binds to its substrate, it lowers the activation energy of the reaction, allowing it to happen more quickly. Let's find out what does the enzyme actually do to the substrate to make the activation energy lower. Some enzymes speed up chemical reactions by bringing two substrates together in the right orientation. Others create an environment inside the active site that's favorable to the reaction. Some enzymes lower activation energies by taking part in the chemical reaction themselves. So that's for today. Hope you enjoy this video and gain a lot of interesting facts about enzymes for your biological knowledge. So please subscribe this channel and click the bell icon. See you in the next video.